Okay, good morning boys and the girl. Today we are starting a new chapter, that is biomolecules. Okay, and this for this term 1, this is the last chapter for your syllabus. Okay, and if you have gone through the syllabus released by the CBSE, there is splitting of the syllabus into two portions, term 1 and term 2. So term 1 exam, as you all know, it will be somewhere in the November, December, and for that up to biomolecules is the syllabus. So as you all know, the name itself says biomolecules. Basically these are chemical compounds that are responsible for biological activities inside us. Okay, so we go with the definition. Biomolecules are chemical compounds found in living organisms. All of us know that through nutrition, through food, when we eat, we get a lot of chemicals in the form of carbohydrates, like whenever you are eating rice, okay, chapati, puri, bread and sugars, what you are consuming chemically is carbohydrates, and they give us a lot of energy, and whenever they give a lot of energy, we are using that energy for various activities. Hence carbohydrates are taken in by all of us in the form of various foodstuffs. Similarly, fats, whenever we consume ghee, oils, sweets, ice creams, we are taking in a lot of fats, and fats also give a lot of energy. Similarly, proteins, whenever we consume fruits, vegetables, soya beans, green peas, white of the egg, milk, a lot of proteins are entering inside us. So when all these enter inside, basically the purpose of we consuming them is to get us energy. So some of them are organic, some of them are inorganic compounds. What do you mean by organic and inorganic? Organic, basically in chemistry you are studying organic chemistry and inorganic chemistry, remember? So when you speak of organic, it is something related to carbon and its compounds. I am sure there is a separate chapter, carbon and its compounds. Inorganic, other than carbon, say nitrogen, phosphorus, sulfur, these are inorganic compounds basically. So whatever we eat, we need to understand what are the chemical compounds that are present in them. Why should we eat them? Okay, many times we said proteins are responsible. When we were talking about ribosomes in the previous chapter, we were talking about the function of ribosomes. We said they are protein synthesizing factories, isn't it? And when they prepare proteins, we grow and develop. For growth and development, proteins are essential. Similarly, what are the chemicals that are present in our cell, in our body? So for that we need analysis. How to do this analysis of chemical composition? Take a leaf, grind it, grind a living tissue like vegetables, in case of animals, piece of liver, like that, in trichroacetic acid, okay, to get thick slurry. Stain this through a chisel cloth or cotton to get two fractions such as filtrate and the retentate. Yes, someone is talking. Please mute yourself. Yeah, mute yourself. Right? Right. So basically what is that you have to do is take any living tissue, maybe in the form of a leaf, in the form of vegetables, in the form of juice of the fruits, and in case of an animal, a liver or a skin, and grind it. in a medium and then use a filtrate to filtrate. So in the filter it contains biomolecules, molecular weight less than 1000 Daltons. Now see that this is how you have to do. Even at home you can do this. And the retentate, that is the top one, contains macromolecules, means big molecules. Macro means big. That's why molecular weight is greater than 1000 Dalton. Dalton is the unit of molecular weight. Just like you say gram, milligram, like that, right? So you are getting a filtrate consisting of micromolecules, means small sized, in terms of weight less than 1000 Daltons. Whereas the upper portion, after the filtrate is down, the upper portion which will be retained in the cotton cloth, that's called retentate, that will be having macromolecules, big sized molecules, whose molecular weight is more than 1000 Daltons. And how about analysis of inorganic compounds? Weigh a living tissue and it is dried. Okay, take a tissue, dry it, and all the water will evaporate out. Look at that. So what is required is, you need to take a tissue, just sun dry it. All the wet portion, water portion will be out. Then it is fully burnt to oxidize all carbon compounds. Why? Why to oxidize carbon compounds? Our aim is to analyze inorganic compounds. That is the purpose. Understood? So it is fully burnt to oxidize all carbon compounds to gaseous form, that is CO2 and water vapor, and it forms ash, budi, powder. And now analyze the ash. The ash contains inorganic elements such as calcium, magnesium, sodium, potassium, and inorganic compounds such as SO4, PO4, NaCl, CaCO3, etc. Now, when we compare the elements present in non-living and living, okay, look at that. Hydrogen content, for example, percentage of weight in terms, okay. Hydrogen, it is 0.14 in that crust, whereas human body contains 0.5% of the weight of the body. Carbon, 0.03 in that crust, whereas in our body 18.5%. And look at the oxygen, 46.6, whereas in our body 65%. Nitrogen, very little, and in our case it is 3.3. Sulfur, 0.03 in earth crust, in our case 0.3. Sodium, 2.8, and in our body 0.2. Calcium, 3.6, whereas in our body 1.5%. Magnesium, 2.1, and in our case 0.1. And silicon in earth crust is highest, silica sand, 27.7, whereas in our body it is negligible. So when we have this comparison, then we understand what are the various micromolecules that are present. So already we have said that micro means small molecules, small sized molecules. And what is their molecular weight? We said just now below 1000 Daltons. Now here we are talking of 18 to 800 Daltons, right? And we need to understand, since we are talking of the living cells, we need to understand what are the chemical compounds that are very useful to biological activity present in the cytoplasm. So we need to understand the cytoplasmic composition. So what are all those present? First and foremost is amino acids. Okay, amino acids are the monomers. I said, what are monomers? Small units. And when the small units join together and form a chain of amino acids, then we say polymer. And the chain of amino acid is protein. So if someone asks you what is a protein, you should say combination of more than one amino acids. Like amino acid 1 combines with amino acid 2, that's a protein. Amino acid 1 combines with amino acid 2, combines with amino acid 3, that's a protein. Okay? So basically our cytoplasm consists of amino acids. Second, lipids. Lipids means fats. And third, carbohydrates, sugars. Right? And fourth, already we have studied in while talking about DNA, nitrogenous bases. So let us see one by one how the structure will be and what is the use of that. Now,
Sir,
yes, please.
above slides.
Above slide. Yes.
Above system.
Yes.
Sir, presence of oxygen percentage, sir.
Percentage. Yes, yes. 1 minute.
Sir, in earth crust also oxygen will be there, sir, inside down.
Yeah. Oxygen in earth crust you are saying 46.6 and in our case 65%. It is in terms of weight. In terms of weight. Okay. And oxygen, as you know, will not be in the form of either atom or molecule in the earth crust. It will be a combination like NO2, SO2, CO2, right?
Ore formation.
It is the form of compounds. Of course, you know, when oxygen atom, your chemistry teacher must have told you, if it is present only in one atomic state, we call it nascent oxygen. But you know, again, your chemistry teacher must have told you that all these elements are unstable. Okay? And why they are unstable? You know that electronic configuration. They must have told you the arrangement of electrons in the outer shell, isn't it? Like 1s2, 2s2, like that. So basically you must understand these are unsaturated. They are not saturated. So they keep on combining with any other element when it is available. So when they combine, already you know in chemistry, either they will be sharing the electrons, okay, or donating the electron. Third option, accepting the electron. So based on that you have got, you know, covalent bond, coordinate covalent bond, like that. So through bonding, the atom join together and make molecules and compounds. That you must remember, right? Now going ahead with the first one, amino acid. So this is the basic structure of amino acid. So centrally there is a carbon atom, as you are seeing, right? And you have got NH2 group. That NH2 group is called amino group. Let me expand that. Yes. I hope you are able to see. Amino group is, you know, ammonia, NH3. Here we are removing one hydrogen, NH2 amino group. And of course, on one side you have got hydrogen, on the other side you have got carboxylic group, COOH. So in this, if you carefully observe, what is keeps on changing, what is that keeps on changing is the R. Again, in chemistry you must have learnt, R is the functional group. Means that group keeps on changing. It can be sometimes NO2, it can be SO2, it can be H, it can be OH, like that. So that decides the properties of the compound. What is fixed? Amino group and carboxylic group, right? And again, carbon 6 electrons, 6 protons, C6, 12 you write. 6 is the atomic number, 12 is the atomic mass. And when you say this C6, these are arranged in the form of orbitals. The electrons are arranged in the orbitals, and I need not tell you, carbon will have 4 lone electrons. 4 lone electrons. We call this as valence electron. Valence electrons. So we say valency of carbon is 4. So only carbon can form easily 4 bonds. So first bond it is forming with carboxylic acid group COOH. Second is the functional group R. Third is the amino group NH2 group, and fourth one is hydrogen group. Out of this, hydrogen is the weak link, right? And this is how the carbon will be forming here. You can see the R group, presence of carbon. So the basic structure of amino acid is this. So one carbon atom centrally with the tetravalency. You say tetra means 4, valency lone electrons, right? So these are the compounds. Amino acids are the compounds formed of an amino group. We put dash NH2. That dash indicates it is unsaturated. And an acid group dash COOH, right? And one hydrogen atom. And as I said, variable group R. It is denoted as R, or it is also called functional group. So amino group and carboxylic acid group are attached to the same carbon atom, alpha carbon atom. So they are called alpha amino acids. They are substituted methanes, right? Now basically again, what is the formula of methane? Methane is one carbon, right? CH4 you write. So you are centrally putting one carbon atom and all the 4 sides hydrogen, 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 CH4, methane. Now here what's happening in this amino acid? One hydrogen is retained, 3 hydrogens are displaced. One hydrogen removed and amino group has come, NH2 group has come. Another hydrogen is removed and carboxylic acid group has come. Another hydrogen is removed, in that functional group R has come. So basically this amino acid has come from methane. Methane, by losing hydrogen atoms, are forming this basic amino acid structure. I hope I have made clear. Yes, Naveen? And I said R is the functional group. R can be anything. Suppose R is equal to H. Then what happens? It becomes amino acid called glycine. So what is the formula of glycine? Basically carbon atom in the center. One side as usual COOH, carboxylic acid group. Second group is amino group. These two are fixed in amino acid. And third one is hydrogen. And R group is replaced with hydrogen. Then the amino acid becomes glycine, very important amino acid for us. Second, if R is equal to CH3, then the amino acid is called alanine. So today after my class, you have to draw all these pictures, practice these pictures and put a thumbs up in the WhatsApp group. And third one, if R is replaced with CH2OH compound, then the amino acid is called serine, right? So if you carefully observe, what we are doing is that R we are replacing. That's why I said it is functional group. Now further types of amino acids. Based on the nature of the amino acid, 20 types of amino acids are used in protein synthesis, and basically the amino acids are of 3 types. Acidic amino acids. The other day we were talking about pH, right? Negative logarithm of hydrogen ion concentration. And I said there will be a scale in pH. The scale ranges from 0 to 14. 0 to almost 7, 6.9. We can consider it as acidic in nature. Exactly 7, neutral. 7 above to 14, alkaline or base. So here the 3 amino acids are based on that. Acidic amino acids, they are acidic in nature. pH will be between 0 to 6.9. Basic amino acids. So 7 to 14 pH scale, they are basic amino acids, and 7 neutral amino acids, and you can see the structure also. Okay, why Prajwal is waiting, not coming, why? Shivras are waiting in the lobby. Okay. Right. Let me see whether room is locked. Okay. So basically, there are 3 types of amino acids: acidic amino acids, basic amino acids, and neutral amino acids. And some amino acids are aromatic. What do you mean by aroma? Nice smell is called aroma. Aromatic, like tyrosine is aromatic amino acid. Okay. What happened? It's not moving. Yes, Naveen. It's stuck, I think. Yes. I think there's some problem. Yes, Naveen. Harshit. No, it's not moving. Okay. What we'll do? Okay, I'll share this in our group. Okay. I'll share this in the WhatsApp group. There seems to be some problem. Just wait. Let me put my mobile data not working. Router 1 is creating problem. Now boys, it's not moving. I think I'll post it in the WhatsApp group, right? We will end the session today. Tomorrow we'll talk. That's also not working.